This is Late Night Science. Today with your hosts Dr. Axel Schumacher and Dr. Darrol Baker. Our guest today, a famous geneticist and molecular engineer, studied at Duke University, now a professor at Harvard Medical School, co-founder of the HLTH Network, who successfully sequenced mammoth DNA. He helped initiate the Human Genome Project and now works on age reversal. The father of genome sequencing, George Church. Hey, it's you. How are you doing? Huh? Look, I'm just polishing some fossils. Have a closer look at this beautiful column of Orthoceras. They're extinct now. They're also called straight-shelled cephalopods. Striking creatures. They rest inside of a dark limestone. Here you can see this here, frozen in time. I mean, over millions of years, their shells, these ones, they're turned into calcite and fossilized. This one was found in Africa. But of course, when those animals lived, well, there was no continent really called Africa. They're really old. I mean, really old. Computer, do we have something on this one?
Yes, I have. Orthoceras are dated to the lower Ordovician to Triassic ages, 500 to 190 million years ago. These specimens are estimated from a time 420 million years ago.
Wow. That was many million years before even the first dinosaurs appeared.
Correct. At that time, the Earth was mostly covered in water. The animals lived inside of their shells, had tentacles they could use to grab food. They had a soft squid-like body. Unlike many crabs who leave their small shells to grow bigger ones, an orthocera didn't have to go anywhere. It simply grew its shell bigger. It did this by creating a new dividing wall inside the shell called a septa. Today, you can see the septas separated by lines in the fossil. Scientists can tell the age of the Orthoceras by looking at how many lines there are. Parts of the body were inside a tube that runs the entire length of the shell, through each of the chambers. Once filled with water, the animal could force the water out, propelling itself backward with a kind of jet propulsion.
They were the ancestors for today's Nautilus family that includes octopus, squid, and cuttlefish. But they went extinct around the time of the dinosaurs, 65 million years ago. I think most of us would agree that it really sucks to get extinct. I wonder if future scientists, many million years into the future, will find our human remains in similar stones. I guess aging and death is a physical process that will always be with us, although nobody really wants to die, at least not too early. Do you remember Aristotle, the great Greek philosopher and polymath? He talked about this already. In his book, De Anima, on the soul, he speaks of the vital principle, or the distinct characteristics of living beings, in contrast to inanimate matter, like this one. Aristotle contemplated the question, what is life and how can it be maintained? And whether death can be avoided, can we live longer? The question is, can we learn something from the animal world and extinct species to help us survive? Huh? Aristotle carefully investigated the possible causes of longevity and mortality. For example, he compared the lifespans of various animals and plants, various lifestyles and environmental conditions. Another Greek philosopher, prodigious physician Galen, in his treatise Hygiene, written around the year 175, framed aging as a natural process that can be eased or even delayed by preventive measures such as a diet. He wrote his books at the peak of his career as physician to the Roman Emperor Marcus Aurelius. Galen thought of aging holistically and recognized that a person's aging path is highly individual, with a wide range of possible outcomes. He developed treatments based on herbs and spices, which he administered to fellow doctors for verification. So in some ways, he was one of the inventors of modern medicine. Generally, advice on how to stay healthy during aging should not be based on opinions and traditions, but on scientific evidence. Yes. Maybe we can learn from extinct species. Maybe not as old as this one, because there's nothing we can extract here on molecules from this. Genetic material and so on. But what about mammoths and Neanderthals? It should be possible to extract DNA from them, at least few fragments. I should discuss this with someone who's actually researching aging and age reversal. Oh, I know. Let's have a chat with my good old colleague, Professor George Church. Computer, do we have anything on George?
George is Professor of Genetics at Harvard Medical School and Professor of Health Sciences and Technology at Harvard and the Massachusetts Institute of Technology. George is widely recognized for his innovative contributions to genomic science and his many pioneering contributions to chemistry and biomedicine. In 1984, he developed the first direct genomic sequencing method, which resulted in the first genome sequence. He helped initiate the Human Genome Project and the Personal Genome Project in 2005. George invented the broadly applied concepts of molecular multiplexing and tags, homologous recombination methods, and array DNA synthesizers. In 2015, Church and his research team at Harvard successfully copied some woolly mammoth genes into the genome of an Asian elephant.
Perfect. That's our man. Let's go. Welcome, George Church. Hey, how are you doing?
I'm great. It's great to be here. Thank you.
Great to have you here on our podcast for several reasons. Because obviously, in the science field, in the genetics vertical, you are a legend already, and I'm sure you will do so much more in the future. So much we can also work together, because what people don't know, you joined Chivom, which is also my company, and we will work a little bit together on making great things in science, like really doing the next level thing. So welcome on Shivom, and welcome on the podcast.
Yes, thank you, George.
Yeah, so people who do not know you, and I guess there's no one out there, especially listening to the podcast, like in a few sentences. So what is your current job there? Where are you sitting? And what are you currently, what's your hottest project?
So I'm a professor at Harvard and MIT, specializing in biotechnology development, including reading and writing and editing of DNA and anything that you can do with DNA, ranging from information encoding all the way up to organ transplants and ecosystems.
Fantastic. So let's jump right in. So one topic I really would love to discuss with you, because I have also a background in longevity, aging. So I worked on late onset Alzheimer's disease and frontal dementia before. And I know you are also fascinated by these topics, working on specifically age reversal. Can you tell our listeners a little bit about what this is about? What is the fascination about age reversal, and what are you working on?
So aging affects almost every source of human morbidity and mortality. Maybe it's not the only influencer, but it influences all kinds of things, ranging from COVID-19, where there's a huge age effect, to even probability of falling down, or the probability of getting up afterwards. That's all age related. So then you have a choice between longevity and aging reversal. The problem with longevity is it takes decades to prove your point to the FDA, EMA, whatever organizations, and to do the experiments, the clinical trials.
Yeah, you run the clinical trials over 100 years, which is probably
or even 20 is prohibitive.
Yeah.
But with aging reversal, you can see effects in weeks, and that's the goal. So it also, a third reason is it has a strong possibility of being something that we all need. Certainly we all do die of aging if we don't get, something else doesn't get us first. And so it could be a big market, which means a low cost, meaning because the fixed costs of R&D are amortized over a large population. So those are the attractions of aging reversal in particular. And what we've done there is two categories of aging reversal. There have been many different methods and pathways shown for animals, but we've been employing two categories. One is what we call cell autonomous, meaning that it only works in the cell to which you deliver the therapy. Things like telomerase, or the transcription factors that Shinya Yamanaka got the Nobel Prize for reversing aging all the way back from 80 years to 0. You can use that in limited doses to get moderate aging reversal in mice. And we just co-authored a paper with David Sinclair's lab in Nature showing that could be applied in a AAV gene therapy delivery scenario. And it worked very well, considering that it only affects the cells you deliver it to. The other, I think, is even more exciting and broader, in that you deliver it to some cells and they have impact on adjacent cells, or even sometimes the entire body. And so we call it cell non-autonomous. It spreads from the cell you deliver it to. And we tested about 45 such gene therapies, we, in this case, being Noah Davidson as a postdoctoral fellow in my lab, and then later he and Dan Elvers and I co-founded Rejuvenate Bio, a startup company, to do the same thing. And we've done it in mice. We've now done it in dogs. The dogs is intended to be a veterinary product in and of itself, but also a segue into human clinical trials that we hope will be starting soon. But of those 45 genes, we whittled it down to 3, and various combinations of those 3 seem to handle a very large number of different diseases that have little in common other than they're age-related. And these are things like type 2 diabetes, osteoarthritis, kidney and heart failure, mitral valve disease, and neurodegenerative diseases. So we're encouraged to think that may be getting at the core of aging rather than at some symptom where you're just dealing with a very specific symptom of a very specific disease. And so that's where we're headed with it right now.
Wow. So do you think you can actually live long enough to live forever, as somebody once said?
I think that's a very challenging question. I mean, I think the first step is to get aging reversal working well, and on all of your systems to make sure that you don't just fix the heart, because then your brain fails, and that's not good. Now, forever is a very long time. I mean, physicists will tell us there's going to be, eventually all the stars will go out. But I think we just want to get close enough to the future that we can see better and we're not in some incredibly desperate race to just survive.
We get a lot of things to do with AI and AGI and transients and things like that. So we get lots of kind of really interesting perceptions into where the biological and genetic meets the AI and the computational, and it's kind of interesting the different types of perceptions that the public have about watching some movie versus the actual reality of existence and time.
I'm a bit of a skeptic about a pure AI strategy. I think a hybrid strategy has certain attractions. I mean, I'm already a hybrid because of I have access to all the information in history pretty much on my cell phone. But in terms of some core capabilities, the human brain is, in the best scenario, in the best established historical scenario, is far better than anything that the computer has produced. I mean, just look at Einstein's 1905 5 discoveries, each of which was worth a Nobel Prize. I mean, that's hard for a human to reproduce, much less a machine. And the biotechnology improvement curve is on a steeper slope. Now that may be temporary, but it's been in place for a couple of decades, where it's going faster than Moore's law. And also, it's better at making atomically precise components. So it's hard to get microfabrication below 3 nanometers, while biology has been doing sub-nanometer engineering or engineering-like things for millennia. So
that's a good point. I think what a lot of people in science do not understand very well, in the public even less, is that nature can do fantastic things. It just shows us really the way. It doesn't matter if you're doing some strange gene therapy things with CRISPR. This is nothing that was invented purely in the lab. It's something copied from nature, right? So nature can all these wonderful things in very small detail, unbelievably quick. Now, the same with the PCR reaction. So it's for doubling from DNA molecules. And I think one of the biggest things we can do as a scientist is take this technology, call it technology that is already there in nature, and use it for our purposes, right?
Absolutely. It would be very hard to engineer CRISPR or anything like it from scratch, or DNA polymerase to make it from inorganic materials the way computers are made. It would just, because in enzymes, in these protein and RNA catalysts, a 0.02 nanometers matters. In other words, you can adjust things by 0.02 nanometers, and take advantage of that. You can change catalytic rates by hundreds of fold at that kind of scale. And so that makes everything more compact, more cost effective, faster, and so on. So I'm pretty upbeat about either biological transcendence or some kind of hybrid where, maybe computers are a little bit better at math or storing, interfacing with. Even biology is even pretty good at storing information in the form of DNA. It's about, a million times higher density and longer life and so forth than purely inorganic.
But nature had billions of years time to develop all of these systems, while we are relatively new to the playing field, right?
Yeah, and you might say that nature was random. But the fact is, even good engineering has a big component of trial and error. Yeah. So trial and error in a certain often will give us a better final product than pure theory alone.
Yeah, that's right. Yeah. But coming back to the age reversal, so I'm interested, how do you want to actually measure age reversal? How do you measure that?
Well, so there are various biomarkers, which I think are convenient for academic experiments, but the ultimate measure of age reversal is functional, medical, and so we've adopted these 8 different disease categories that have nothing in common other than that they are age-related diseases, and so if you can decrease the probability of succumbing to those diseases. You can artificially induce the disease, or you can let it naturally occur in a pre-aged animal or pre-aged human. That's the bottom line. Is you want to not one disease, because you do one disease and you could have overfit your functions to just deal with that disease. But if you get multiple diseases, then you're probably on the right track.
Right.
That's our measure.
In this context, I'm not sure. What do you think about so-called epigenetic clocks and to use them to measure the aging process? Let me just explain for our viewers what I'm talking about. Let's imagine like we have a DNA strand. This is our base pairs of the DNA on one large strand. What we have there is what can happen, that so-called epigenetic marks of DNA methylation patterns. They can attach to the DNA on different points on our DNA and switch our genes on and off. So these are specific patterns. We can measure these patterns easily nowadays. But sometimes what happens during the aging process, something can go wrong. And then you have maybe a wrong epigenetic mark at some point. And again, we can measure this. So what happens is a lot of scientists, and which is also actually based a lot of the work I had in this field, also on Alzheimer's, we can identify these patterns and see if with age something changes. And of course, if we do then apply age reversal, let's say in a model animal system, we can then measure accordingly, if we look at these patterns, if the aging process goes back to the younger state, or if it progresses or is at a steady state system. And of course, we can measure it for epigenetic therapies, meaning we can maybe remove certain epigenetic marks, which switches then genes again on or off. What do you think about these methods, the epigenetic application in aging reversal?
Well, essentially everything that we do in our lab is related to epigenetic therapies. Whether they're cell autonomous or cell non-autonomous, whether they spread or not, they're epigenetic. And that's in contrast to another school of thought, which is that aging is about damage and you need to fix the damage. Our theory is that if you fix the epigenetics, the young cells will take care of the damage, as they do, in children. So we're very big on epigenetics. We do measure epigenetics occasionally. Like I said, the biomarkers, which epigenetic biomarkers, are useful in academic research. But we know as synthetic biologists, many of those markers that you showed on the DNA, you are not causally related to a disease. They're not causally related even to a gene that seem to be involved in the disease. And so that means they're literally markers that are correlated but not causative. And that means that we could, but synthetic biologists, clip those things off and nothing will happen. Conversely, we could fix the epigenetics, so, like with the Yamanaka factors, just set it back to 0, and then set those epigenetic marks back as if it were an old cell. And so I think that because they're not causally related, we want something that is on the direct path to what we care about, which is medical outcomes.
So basically, you want, again, use our cells' inherent ability to repair itself to get us back to a younger state.
To the extent that repair is the problem. I mean, I think there are many other problems that is caused by being in the wrong epigenetic state, meaning old epigenetic state, that are not necessarily due to damage. In fact, you could have a completely pristine genome, no DNA damage whatsoever, and still die of old age, because that's how powerful the epigenetics is. But it isn't necessarily related to these marks that everybody uses as their clock. It's a great clock. It's precise to about plus or minus two years, but it isn't causal. And that means that you can, as you start messing with aging, you can get a disconnect between the non-causal biomarkers and what you actually care about. So we just have humility as synthetic biologists that we know we can disconnect these two, so we better have to be careful.
Right. But that brings us back to an old question of evolutionary theory. Is aging pre-programmed in our
Almost
DNA?
Certainly. Almost certainly. You look at a mouse lives about 2 years, even if you protect it from predators, and a bowhead whale lives about 200. And now, admittedly, there's a lot of differences between them, but it's clear that generation after generation, the whole family dies at 2 years if they're mice, and the whole family lives, 150 plus years if they're whales, or bowhead whales. So that's fairly likely. And then these experiments that show aging reversal also indicates there's some program there, so where you don't have to change the DNA, you just have to change the way the DNA is regulated.
Right. Darrol, you wanted to ask something there?
Yeah, George. I mean, you're a very, very famous vegan as well, so it's kind of interesting to get your thoughts on Bill Gates' advocacy of synthetic meats and how to avoid climate disasters he talks about. But it'd be interesting to sort of get an understanding of one aspect of this is would a vegan actually eat synthetic meat? I know that sounds a bit strange, but
no, it's a good question, because it depends on why you're vegan. There are many reasons that I could list, some less obvious than others. So there's animal welfare, which is one of them. There's which the clean meats, the vegan meats does solve. There's zoonotic diseases like COVID-19, swine flu, so forth. There's the antibiotics that one needs for modern meat production. There's resources like sun and water that require maybe 20 times more resources, and sometimes fossil fuels also to produce meat. And then there's health. And my journey into veganism did start with health. My father and I have a cholesterol problem, and he died as the consequences of a triple bypass and cognitive decline due to low oxygen. So, and that would not be solved by the clean meats. Like Bill, I am working on clean meats. I'm working with a couple of companies like Memphis Meats to produce these, but it's not yet absolutely clear that they can be produced for less cost than plants. And plant-based meats, like Beyond Burger, for example, is an absolute terrific tasting thing. I have a lot of my carnivore friends who like it better than regular burgers. So we don't need to go to clean meat to solve the global problem. In fact, there are quite a few people that are basically vegan because they can't afford anything else. There's some people who can't even afford vegetables that are good enough to give them vitamin A. And so 1 million people a year die of blindness due to vitamin A deficiency. And one of the solutions that's been now approved is golden rice, which is an engineered form of rice that has it's so much vitamin A, you can see it in the color of the grains of rice. So we need solutions. I agree with Bill, but we need solutions. I think a lot of them are going to come from more efficient crops, more efficient use of land that isn't currently fully utilizing. A lot of it is dedicated to meat production when it could be dedicated to food.
I agree.
3 times more efficient food.
Yeah, I agree, because I kind of think that if we've got a population that's rapidly increasing over the next 150 to 100 years or so, the implications to the population and how land's used to feed people, is it sustainable? And so to me, it's kind of interesting what he talked about, and the amount of less resources that we needed to actually produce it was kind of fascinating for myself. I didn't really think about it at the time until he actually brought those points up, but I was thinking more about crop erosions or soy-based plants for cultivations and instead of cash crops or biofuels being replacing for cash crops and that kind of stuff. I didn't really think about it in terms of feeding the world's population, and it was a sudden realization that there is a dichotomy between these two things, and genomics can answer that question too. But as you say, transgenics or transgenomic crops and things like that, how we can actually do that.
But transgenic crops is we have a problem in the public perception. I think that changing natural products is usually seen as negative. What do you think, George? How can we deal with this?
It's all relative. So it's whether the benefits outweigh the risks. And even if the benefits are low, then the risks have to be zero, which is almost never achieved with any realistic technology. And I think the thing is, there are many GMOs for which there's very little discussion. There's very little worry. For example, moving the insulin gene from a human into a bacterium and then massively producing millions of liters of bacterial-produced insulin, that's fine. That's a GMO that every diabetic in the world uses, and all their families and friends are very supportive of it. The problem with GMO foods, tomatoes and strawberries and things like that, is they don't provide much to the customer. And much of the original pushback was from wealthy Europeans, who particularly don't see an advantage. And they see the disadvantage of an economic disadvantage of having a monopoly like Monsanto from the United States, impacting a European economy. And so I totally sympathize with that point of view. If it doesn't help you much and it has a potential economic impact, then you should push back. But things like golden rice, GMO insulin, that sort of thing, there are human lives at stake. And I really think that that's a completely different issue.
Yeah, I agree with that.
The same thing probably goes for climate change. There's a lot of human well-being and lives at stake. And so a little bit of well-placed and well-tested engineering could have a much bigger impact than switching our lights from tungsten to LED, or switching from SUVs to bicycles. I mean, even if the entire planet of humans stopped using fossil fuels and behaved very well, there still is that's 10 gigatons of carbon. That's only 10 gigatons of human carbon, but there's 1,400 gigatons at risk just due to the natural processes that were initiated by our ancestors when they killed off all the herbivores in the Arctic. So we need to be sequestering carbon, not just not putting more up there. So most of the proposals, anyway, I think that there is some fairly low cost, low risk methods of using engineering to reverse climate change, not just slow it down.
That's a perfect segue to my next question. Your talking about the carbon footprint. And you mentioned already that humans probably also played a big role in the extinction of certain animals, like the woolly mammoth.
Yes.
And one of your projects that you started several years ago is to bring back the woolly mammoth. Can you explain a little bit what this project is about?
Well, it's more about not so much about bringing back an animal species, but saving endangered species, the Asian elephant, and also restoring a vibrant ecosystem in the Arctic. So the Arctic used to be a beautiful grassland that was very good at sequestering carbon. Each growing season would add another layer of roots until it was 500 meters thick, which is about 500 times thicker than most of the tropical rainforests. And so most of the carbon in the world is stored in the Arctic because grass was so good at it. But when the herbivores got extinct due to human and other forces, that changed from a beautiful grassland into these scraggly trees that are very dark, and they bring in heat through their albedo, their darkness. And then they also they allow snow to pile up, which protects the -40 from getting down into the soil. And so the warm soil stays warm because of this downy blanket. And then the grass is better at photosynthesis. For those three reasons, a restoration of that fairly recently lost, maybe in the last 10, 20,000 years, restoring it back to that would have huge benefit, not just for the ecosystem, but for human efforts to sequester carbon. And this could be done with very low or maybe zero belt tightening for the consumers. It's just encouraging a natural process in the world rather than having small effects due to changes in how you use energy and transportation.
Right.
I'm all in favor of consciousness raising via all of these human carbon footprint related things, but I think much bigger than our carbon footprint is what we can do by sequestering carbon and reversing back to pre-industrial levels.
But we still have the technical problem. Let's dive a little bit deeper into that.
Yeah.
Obviously, just bringing back extinct species is extremely difficult, if at all possible. We don't know that yet. So far, we haven't really brought back anything.
Well, we brought back 2 viruses. That's not really a mammal, certainly.
I think we have enough
we
of
brought back 2 frozen mammals. A black-footed ferret was 32 years frozen, and a horse that was frozen for 40 years. Problem is, mammoths were frozen for a lot longer than that and under less than ideal circumstances. So we probably can't do that kind of de-extinction event, but we can do it. What we can do is something maybe even better, which is we can make a cold-resistant elephant that has, dozens of genes from the woolly mammoth. So we've de-extincted the genes rather than the species. Plus, we can add in genes that protect elephant against modern problems, like there's an extinction-level virus called EEHV. It's killing off the baby elephants. We can make them resistant to that. We're getting very good at making, either by vaccines or CRISPR or something like that. And we can also make them poacher-resistant. That sounds funny that you could genetically engineer an organism to be poacher-resistant, but it turns out we know how to make tusks either very long or very short. So you can make them much long, many times they're the size of their head, or there's natural variants that occurs occasionally in natural populations where they're almost invisible. And so you can make it so you could dial it up and down. So if they're out in the wild where they could be poached, they have short tusks. And if they're in a well-secured, protected environment, they could have long tusks. And so I think those 3 things, so those cold resistance, virus resistance, and poacher resistance, is better than just bringing back the mammoth. It'll look like a mammoth, it'll behave like a mammoth, it'll love -40 degrees, but it'll have all these genetic advantages over current Asian elephants. They will basically be an Asian elephant, so we will be preserving that species as well. A very close relative to the Asian elephant. It would be a hybrid.
I think it's called proxy species, right? So you are basically mimicking what's already or what was extinct.
Right. It's a very close proxy because the mammoth and the Asian elephant are very, very similar. Right. So, for example, it's not like a proxy species where we bring in a polar bear or something, which is a carnivore, not a herbivore. Yeah, it's much closer. In fact, baby elephants often are covered with woolly hair, and when you have elephants up in Canada, they love to play in the snow, and they will go and they'll break into the ice of the lakes and swim in the cold. So they really are very close to being mammoths. They just need a little nudge.
But to bring them a little bit closer with genetic technologies, I mean, I saw some photos of you where you were basically, I don't know if you dug them out yourself, but you got some bones back from
yes, those I got myself. Yeah.
Oh, cool.
Actually, it was a combination. The frozen ones, other people got, but the non-frozen ones I got myself. Yeah.
Fantastic. That must be fun. But how much DNA can you still get out of those?
You only need trace amounts of DNA. So, we can, in humans, we can sequence DNA from a single cell and get a pretty accurate image of it. For ancient DNA, it's hundreds of times less efficient, but you still only need, a few, fingerfuls of sand-like tissue. So that's not a problem. And then you get it into the computer and you reconstruct it and you compare it to elephant DNA, and you can see the differences. And there are very few differences. It's like 3 differences between elephants and mammoths that give you blood that's very cold tolerant, and that's been proven. The gene has been resurrected by the Campbell group, and it's been shown to be cold resistant. Mm-hmm. So we know those processes. We know how to get the ancient DNA, how to sequence it into the computer, and then go from the computer to fresh synthetic DNA, and then test that gene provides cold resistance. And there's another set of genes that provide cold resistant nerve endings, and so on. We're getting deep knowledge of what causes the fat containing lumps on their back and head, fat all over their body, the thick long hair. All these things are things that are known both from studying elephants and other species. So that's making great progress.
Well, how long does it take? I mean, obviously, elephants, they are not grown in labs like mice, so it takes probably quite a long time to really adjust or modify their genome to the environment. Well,
interestingly, the modification of the genome can be very quick because we don't have to we don't have to go through a whole elephant generation to test it. So we can, many things, the 2 things I mentioned already can be tested in the laboratory with elephant and mammoth genes side by side, but without making a whole elephant. But even if we make a whole elephant, a lot can be tested, on a 2-year-old elephant. You don't have to wait until it's, they live to 60 years old. You don't have to wait that long. They are, the females are reproductively fit around 9 years old. The gestation period is 22 months. Now, we've kind of de-risked this whole process as well. You can do very similar experiments in pigs. And so we've now got pigs that have been engineered at 42 different genetic locations. So 42 changes, which is a lot. Typically, like when you're doing human genetic engineering with gene therapy, that's 1 gene at a time, or maybe 2. But in pigs, we've done 42. We call it pig version 3.0. They're intended for organ transplants, and we have about 2,000 of them now. So that's kind of proof of concept that we can make herds of these engineered organisms. And another precedent example is the bison. The bison was down almost extinct in the wild, and now we have 500,000 of them, 500,000 worldwide. So it shows that we can really grow, once we have the right genetic material and the right plan, we can put them back in the wild.
Wow, that's fascinating. But I wonder, so, I mean, you almost, I guess, like 80% of your life, you dealt with biology, with looking at our core, on our DNA, what we are made of. Do you think that the development of life on Earth is very specific to our planet, or can a similar system have evolved somewhere else? I wonder.
Well, the answer is an open question. It's a question that can be answered in a variety of ways. I mean, we're looking for signals, looking for planets that are Earth-like and have atmosphere that are consistent with life. And that type of study is getting better and better every year. We're also simulating it in the laboratory. We're asking, how special does the environment have to be and how fast can it go? So if it can go within sort of the time of a PhD thesis, then you've got accelerated evolutions, accelerated origin of life from inorganic materials. So that's another line of research, but neither of those have yielded anything that's convincing enough that I think we need to consider the possibility that we're the only intelligent life in the universe, possibly the only life in the universe. But it could be just the opposite. It could be we're completely surrounded by alien life. It is one of our most embarrassing gaps in our scientific knowledge is whether we're alone out of, trillions of stars, whether we are completely surrounded and we don't even see it.
Right. But on the other hand, I mean, one thing that obviously you are an expert on is on synthetic biology, basically building something from scratch. So where are we going with these things? Can we design life in the Petri dish and come up with something completely new that we have never seen before?
Well, this is challenging. Most of the synthetic biology are small variations on existing genomes. Deleting a few genes, rearranging them. Probably the most radical change is what we've been doing is changing the genetic code, where every gene is changed in multiple ways. That still is very closely related to the original. Probably the thing that will come closest to something really new is this origin of life type of study, where you're really starting from chemistry, and you can either start with the chemicals that we think existed on early Earth or early universe, and then go from there, or we can just use a whole other set of chemicals that we think could also evolve life, and those are two interesting pathways, and they're the most likely ones to turn up something that's not related to the DNA and RNA of our current world. Then there's motivations for making new things that are functionally new, that are based on biology's amazing ability to do this kind of atomic precision engineering, or help us do that. And one of my pet projects is trying to send a new life form, a synthetic biology form, to Proxima b, which is 4.7 light years away, and then have it construct a communication channel back. In fact, I just participated in a breakthrough Starshot conversation on that. That's a great synthetic biology challenge. Another synthetic biology challenge is making cells that are resistant to all viruses or resistant to all pathogens.
Do you think an ancient, reference genome, an ancient human reference genome, would be an interesting thing to get to, in the sense of that could be the origin of? I'm humanist here in terms of bias, but could it be an interesting segue into how diseases evolved and how a variant came about? I know it's hard in terms of creating an ancient record human genome. And again, where would the origin of that be? But equally, it'd be interesting to see how we've started here and got to here, and could actually map out where those mutations are and how they kind of evolve.
No, I think that's an interesting point, and we're doing that in a certain sense. We have the nearly complete genome of humans, of Neanderthals, and of chimpanzees, and we can do that three-way comparison. And we can even correlate the small change differences between us and Neanderthals would probably look like us if we dressed them up properly. But those small changes, we can correlate with diseases, and there are some diseases of us. So many people in the world have Neanderthal DNA. I'm one of them. I have like 3% Neanderthal DNA, which is not a record, but and you can associate diseases with the chunk of the Neanderthal version of that DNA. If you wanted to do functional tests, so you can make highly engineered cells, and those highly engineered cells could even be used in organ transplants. So just like we're making pigs for organ transplants, we are also making human organs derived from human pluripotent stem cells from my left arm. And those organs to be transplanted, and then you can see how they function. So you could, in principle, have an organ that has even more Neanderthal DNA than I have, and see how it functions. I think that will probably happen.
Mini Neanderthal, that would be fascinating. Yeah,
it's if they're perceived to have advantages where they would be resistant to a pathogen or be more robust in some medical sense, then we will probably do it. Also, it's unknown just how much Neanderthal DNA there is in our population, and Denisovans. So there's another ancient species that's about as far away from us as Neanderthal is, but also far away from Neanderthal. And our current population has a mixture of all these. So in principle, just by some marriage arrangements, you could get people that are highly enriched for those two variations and depleted for the modern DNA, and they would probably be fine.
Right. So we in theory, we could partially breed out our new Neanderthal and Denisovan lines back where they came from, right? Yeah.
So there are people who might find that desirable from their family, what their family origins, and I don't think the government is going to interfere with their personal choices.
Yeah, that's probably because there are a lot of those Neanderthals specifically working in government.
Yeah, and Denisovans too.
Oh yeah,
but hopefully they won't interfere either way. They won't make us do it, and they won't stop us from doing it. Right? Yeah. Right, it's a personal choice who you marry.
Which government are you talking about, Axel?
I'm talking about personal choices. So obviously, technology. I mean, everything on our planet changes with our technologies. And so, George, where do you see us as a species developing with all these new technologies, CRISPR, nanotechnology, and how we can modify ourselves in theory?
Well, we're modifying ourselves more than in theory. Like I said, we're hybrids with electronics. We are superhuman in the sense of we're resistant to everything we're vaccinated for, which our ancestors would have killed to get that superpower, to be resistant to smallpox and polio and mumps and measles and all that stuff. A huge fraction of babies and mothers died during birth in our ancestors. So we're superhuman already. But because we're hybrids, because we get so much benefit from phones and from airplanes, and we don't need to be super strong or super fast because we're not going to be stronger or faster than a jet. So we might as well face the fact we're going to be a hybrid. So what's left is really aging and intelligence, cognitive ability. And I think we are changing ourselves cognitively, partly because we have such an old population now. We're terrified of cognitive decline in ourselves and in our parents and friends. And so we're working hard. You mentioned, Axel, that you worked on Alzheimer's. My group works on Alzheimer's and other neurodegenerative diseases I mentioned in dogs. The consequence of that may be we may overdo it. That if we get good enough at preventing cognitive decline, that might be also result in cognitive advancement. And our species has no particular problem with cognitive advancement. That's the whole idea behind, schools and books and that sort of thing, computers. But this would be a little different. This would be something that's more biological.
But we see also the negative effects. I think there were recent studies that showed that we have in recent years actually a decline in the average IQ in younger people, particularly. So, which may be.
Yeah, I think the jury's still out. I mean, there's the Flynn effect where we see an increase in IQ with time. And then there's smaller effects that fluctuate from year to year.
Yeah.
Which, dependent on cultural effects and there's all kinds of confounding effects in IQ tests. And IQ doesn't necessarily even measure what we care about. But, I think we should pay attention, though. I mean, we should, is it nutrition? Is it, failure to vaccinate? Is it, there are many reasons.
It cannot be something evolutionary, right? So I would argue that our ancestors probably
not in the conventional sense.
If you would take some person with a time machine out from 50,000 years ago and would transplant this person into our current time, they will probably do just fine.
They would do fine, but they would probably not be as tall as we are, because there's been an increase in height with time that has to do with the nutrition of our grandmothers and our mothers, and that probably also has something to do with our cognition. And there's some things that take a few generations to kick in. So, time machine will bring people back that are different, but still fine. They would fit into society just fine.
I think it's all about investment in our youth and our youngsters to make sure that there's education and health and activity in many ways and making it so that the planet is better than you leave the planet better than you brought into it, basically.
Talking about making things better, George, do you have any advice for young people who want to go into science? So where do you think they should go? What are hot topics? Like, imagine you would be young, you would be 16, 17 years old, and you would decide, hey, I want to do something to change the world. What area would you go into?
Well, I think all sorts of space-related science is important, especially biological components. There's a lot of emphasis on the physics, but we need a lot more studies on human adaptation to radiation, to gravity, to microbial problems and psychological problems of having close quarters. So that's one field. Another field I think is ripe is applying AI machine learning to medical and biological problems. Like we've published 4 papers on applying it to protein design. Proteins are these are the nanostructures at work. And so that combination of using big clunky computers to help us make tiny, more efficient ones would be quite cool. And then almost anything having to do with synthetic biology is going to be very impactful. Everything we've been talking about in this session, I think is because the rate of change is so steep, and because we have exponential improvement in these technologies. Every few years, you get a level playing field where all the incoming young people have a slight advantage over the old people because they have a fresh mind and they can see how it is, while the old folks are still trying to protect their turf and keep it the way it was, keep the technology so that they're comfortable with it, while young people come in and they have an advantage. So look for fields where there's an exponential change in cost and quality and qualitative changes. And I think synthetic biology is a perfect example of that, reading and writing DNA.
Yeah, very good idea. Yeah. Just before we close, I'm curious about, again, like if you would start again, if you look back at your career, do you think there were mistakes you made? What would you do differently, or what should a young scientist look out for when going out to the university? And should they stay at university? Should they maybe go and start a company instead? I mean, there's so many options.
I think it varies from person to person. You have to make your personal choices. I mean, a lot of young people have to do something from a financial standpoint. So I felt a lot of pressure when I was in college to finish college early and get a job. And the job that I chose was graduate school, but at least it went from me paying the college to the college paying me. And so I finished in 2 years, mainly for financial motivation. And I think that's one of the things that varies from person to person is, how far into debt are they willing to go in order to get a better better education? Now, a lot of education can be achieved on the fly. So I would say almost everything I do now, I didn't have a course in. For example, mostly what I do is engineering. I'm in the National Academy of Engineering, but I never took a course in engineering. I do a lot of computer programming, but I had so much computer programming before I got to college, I didn't take any courses in computer programming. And the list goes on. So I don't want to over or underemphasize the importance of formal education. It's a great place to meet people. So even though, Bill Gates didn't finish and Mark Zuckerberg didn't finish their degrees at Harvard, they did take advantage of Harvard in that they met some very key people that helped them launch, two of the biggest social and economic phenomenon in history. So I think even a year in a elite university can be extremely helpful to meeting people and getting oriented at your priorities. So.
Wow. Yeah. So, George, imagine you got a lot of money. Someone would throw money at you. What would be the next big project you would work on?
I think if you some people get too much money. I'm not saying that I would get too much money, but some people, when they get too much money, then they become obsessed with maintaining it and keeping it away from other people and things like that. So that's one thing to be cautious of. In fact, a lot of lottery winners are some of the saddest people on the planet. That said, I already have some fairly, some hobbies that could benefit from, serious scientific problems. So we've mentioned a lot of them, which is dealing with diseases of aging. That's not cheap, although getting to other planets, ideally even outside of our solar system, is going to take some money. Safely and thoughtfully engineering ecosystems to restore them to a better place and to do carbon sequestration. That's something where a little bit of money could, in fact, all of these are examples where a little bit of money could have a big return. So the question isn't what you would do with a lot of money, it's how you would use a little money to save us trillions of dollars. One of my pet projects is the bioweather map. I've been advocating this for a couple of decades. It's analogous to weather map, where people get engaged. The regular public looks at the weather in the morning to decide whether they're going to, walk or take the car, that it might prevent them from, slipping and falling on the ice and hurting their hip and so forth. The same thing, you have life-threatening events that could be solved by a bioweather map, where you decide whether you're going to go to daycare or to your job based on what pathogens are there that morning, or you can see what's happening on global scale, so you can make decisions, about travel and so forth. So I think we need bioweather map. The cost of that is large, but it's thousands of times less than what we're spending on just one disease. So this would save us from many diseases. Just one disease is costing us $16 trillion in the United States alone. And our preparedness has an impact, because, so for example, China and other Asian countries were prepared, and they had 400 times fewer deaths per capita.
Right.
So we need a bioweather map, and it's cheap compared, it's like, if you think education is expensive, try ignorance. So most of the things that I would spend money, things I am spending money on, are aimed at saving us from, painful, horrible, diseases, and saving us money.
That's a very good strategy. I like that. That's what we do too.
Yeah.
So, Darrol, do you have any last questions or?
No, not really. Only to say that George and I worked together many years ago at Gnome. So it was a long time ago.
Yes, that Gnome lives on in companies like Veritas and Nebula Genomics.
Yeah.
We will not be satisfied until everybody who wants one can get their genome.
Indeed.
And for that matter, infectious disease as well as inherited disease. So thank you for your help with Gnome.
I noticed that we have probably a slowdown in getting genome sequencing adopted for the public. So of course we had with 23andMe coming out, like, I don't know how old they are, 10 years, 12 years. That in recent times it slowed down a little bit. People are early adopters.
I think what has slowed down is things that are ancestry related. But what is picking up is whole genome sequencing, which is much more medically related. And so there are some things that are somewhere in between where you get a little bit of medical information, but what we really want is the whole deal. We want the whole genome. We want a few other omics that have to do with infectious disease and immunology, but that will provide us with much better preventative medicine. And preventative medicine is much less expensive, much less painful, many fewer visits to the hospital.
Exactly.
That's where we're going.
So that means people get sequenced, get vaccinated, and stay healthy.
Absolutely. Yeah.
All right.
Thanks, George.
George, thank you so much. It was
my pleasure.
Talking to you. And I'm looking forward to fantastic projects we can do together in the future.
Yeah.
Thank you so much for coming on our show.
Yeah, sure.
Thank you. And all the best to you. Stay safe.
Yeah.
Avoid the pandemic.
Yeah.
And thank you so much.
Okay. Take care.
You again. The podcast is over, right? But coming back to Aristotle. Aristotle raised a crucial question: the possibility of a radical life extension or even immortality. He believed that any physical entity is composed of opposite and conflicting elements. According to him, any such entity must be in a constant process of change, and therefore cannot be preserved, like this one, as is, for a long time. Thus, a living organism is fated for ultimate destruction. for it contains in itself antagonistic forces and elements that mutually annihilate each other and thus destroy the entire body. This could be epigenetic markers or other molecules that break down. Similarly, Greek physician Galen emphasized that life could be prolonged, but was of the opinion that death is inevitable, as the body deteriorates of itself. In other words, we have to face the impossibility of our body's preservation beyond a predetermined time limit, at least in a living form. As we can see here, the Ottocera are still preserved. To keep it preserved, I will put it back to the shelf where it belongs. You don't have to watch me doing this. You can switch off, right? Go. But before you do, please help me out with this and subscribe to the show and press the like button. And of course, if you watch this on YouTube, please press also the bell icon so that you'll get notified whenever something new comes up from the late night science show. Well, that's it. Good night.